So here at STEManices, we are all about giving you both sides of the story so that you can make informed decisions about what you think about science or different issues in the news. But there has been a recent issue that has come up that needs to be addressed, and we are going to give you...
we need to take a stand here.
Yes, a strong stand. So before we get into what a CRISPR baby is, we thought it'd be best to give you kind of a explanation of gene editing versus gene therapy, since we've already done a video on gene therapy, just to kind of get you up to speed.
Right, so we talked about gene therapy and how the purpose of that is to deliver a gene into an organism where maybe you don't have that gene product. You just package it up, usually using a virus, and deliver it where it needs to go. But gene editing is a little bit different. You're not adding this extra component, you're working with what's already there. So you take the organism's DNA and you basically edit it to delete things or cut and paste, make any kind of change you want.
Mm-hmm. And this is useful for lots of reasons, because you can delete a faulty gene that's not working, you can maybe turn up a gene and make more gene product that's produced. So it has many uses, especially in the lab. We can use it to study the effects of genes.
So there's lots of different gene editing technologies, but the one that's the most popular lately is CRISPR. And this stands for clustered, regularly interspaced, short palindromic repeats.
Hence why it's abbreviated.
Yes. So CRISPR was actually discovered in bacteria, which is pretty cool. It's kind of a way for bacteria to have an immune system and protect themselves against viruses. Mm-hmm. So what they do is they recognize short repeats in the viral DNA, and they kind of save those pieces in their own genome. These are the short repeats that the name refers to. So then the next time the virus tries to infect the cell, it can activate those repeats, they can be transcribed into RNA, and they then work with their partner protein that's a nuclease. So nucleases cut DNA. So this RNA and protein work together to cut up the viral DNA, essentially.
Yeah. So it's basically like storing these sequences in a library so you can access it again and call on the police to go and kill the virus. So it's a really, really awesome system.
Yeah. So this actually was known about for a long time, this kind of immune system. But in 2012, it was kind of simultaneously discovered by Jennifer Doudna at UC Berkeley and Feng Zhang at MIT that we can actually hijack the system that bacteria just made by themselves, and we can use it in animal cells. So we can make our own repeats and guide this nuclease, this Cas9 protein, to specific, precise locations in the genome and make a cut wherever we want.
Mm-hmm. Yeah, so it's like this really awesome Swiss Army knife of molecular biology tools, because you can have it cut and delete, you can have it cut and then something replace what you cut, you can have it turn on a gene, turn off a gene, you can have it turn up basically the volume of the gene or turn down the volume of the gene. So it can do so many things, and it's completely transformed the way that we work in the labs.
Yeah, the possibilities are limitless. But there are some limits on this technology presently. So I mentioned that short sequence that acts as kind of like a address to a specific place. Sometimes that address isn't really specific. We're really restricted in like how long the address can be. So sometimes it makes mistakes and recognizes other addresses that are really similar, and this is called off-target.
Yeah.
So you can make cuts in places that you don't intend to make cuts. And this can be bad because it can cause mutations that in like genes that are really important for you to be able to live. They can cause mutations in oncogenes and cause cancer. So we are kind of worried about that aspect. The other thing that we need to work on with this technology is that it's not very efficient.
Yeah. Yeah, I mean, when you use it in the lab, like, you can get maybe 40-50% efficiency.
Yeah.
But to get 100%, that's unheard of.
Yeah, I mean, it depends what you're doing. If you're trying to delete something, that's really easy. But if you're trying to cut and paste, we're still around maybe 40% with the best reagents and everything.
Yeah. And one other thing with CRISPR too is, yes, this is very useful for us in the lab, but what should we use it for beyond that? Or should we use it beyond that? And just the ethical implications of going in and cutting DNA or editing someone's DNA. And then you get into the question of, oh, should we edit the germline?
Yes. So you know that we have somatic cells and germ cells. And germ cells are the egg and the sperm that are going to go on and make the next organism. Mm-hmm. And somatic cells are something like your skin cell that just affects you. It's not going to go become a person.
Right. So if you edit the germline cells, that's going to affect future generations for years to come.
Exactly. So a lot of scientists feel right now that we should only be editing somatic cells, and because we don't really know what we're doing yet, to make sure that we don't cause problems for future generations. Mm-hmm. The other thing scientists really feel is that this kind of technology should only be used for like really life-threatening diseases, no other cure available.
Mm-hmm. And currently, for editing the germline, it's banned in the UK, in the US, and China, and tons of countries because they recognize that this is not something that we should be doing because we don't know enough about the technology and the effects.
That being said, at the end of November, the scientific community got a big surprise that apparently this technology has been used to make babies already. So they use this technology on embryos in China to make twin babies, Lulu and Nana.
And this came out at an ethics conference about gene editing.
Yes.
For someone who was supposed to give a talk
on the roadmap to proper and ethical gene editing. His talk was canceled. So who is this guy? He's a professor at a university in Shenzhen, China, and the purpose of his project was he wanted to make babies that were immune to developing HIV. So how he did this was by targeting the Ccr5 gene. Mm-hmm. This gene's normal function is that it's a receptor, meaning that it sits on the cell membrane on the outside and it interacts with specific proteins. They fit into this receptor like a key into a lock, and that triggers some response in the cell. But the bad thing about this lock is that it can also be used by HIV to actually enter the cell. So it's been shown that some individuals with deletions in this lock protein actually are kind of immune to developing HIV.
Wow.
So his idea was if we get rid of this protein completely, then these children won't be able to develop HIV/AIDS. And so he recruited patients from an HIV/AIDS clinic in China, promising them that he was working on developing an HIV vaccine that would make their babies immune to this disease. So he was able to successfully edit this CCR5 gene in 2 embryos, and he states that there is 1 potential pregnancy on the way too, so possibly 3. But in the 2 babies that were born, he claims they're healthy, and one of them is homozygous, meaning that they have a deletion in both alleles of the CCR5 gene, and one of the patients is heterozygous, so they only have 1 deletion and 1 of the copies is unaffected.
So we assume that he's done this. He hasn't published anything, so we really don't know, but there's definitely a lot of caveats with this and things that have made the science community very angry at him. One of the biggest things is what's called Mosaicism. And Mosaicism, if you think of the word mosaic, it means like you think of tiles and a bunch of tiles, different colors all together. And that's almost what Mosaicism is for genes as well. So if one of these twins, they said that one of them shows Mosaicism, which means that some of the cells have the copy of CCR5 deleted completely, other cells do not have it. So that could mean on their skin, you could have 1 patch of cells where they're immune to HIV and 1 patch of cells where they're not immune to HIV, and on and so forth. And so this is really, really dangerous just for a lot of reasons, because mosaicism is never good because it has all these off-target effects as well, because if half of your body's responding to HIV and the other half's not, you're almost like warring against yourself.
Right. And the particular cell type that this lock, CCR5, is expressed on is white blood cells. So if you don't delete the gene in the white blood cells, what's the point? You've put these people at risk for no reason. And just in general, scientists kind of agree that the costs really outweigh the benefits in this situation. Largely because although HIV/AIDS is, a terrible, deadly disease, we've made a lot of progress on detecting it and educating people on how to protect themselves from getting it through safe sex, etc. And we know a lot better how to treat it through retrovirals. So although it's horrible, it really is, more manageable. And people argue that it doesn't warrant this kind of gene editing intervention as much as other diseases, such as Huntington's disease, where we don't have a cure.
Right. And with Huntington's disease, too, it would be easy to go in and just delete the gene that causes Huntington's, whereas CCR5 is more complex. And the fact that he deleted CCR5 in these twins makes them more susceptible to other viruses, like the West Nile virus. They're more susceptible to the flu. So, yes, potentially they're immune to HIV, but then you have all these other potential issues, too.
So one of the big things that ethicists worry about this whole experiment is that the parents weren't really given informed consent, meaning that it wasn't really explained to them exactly what was going on. I mean, we don't have the consent form in front of us, so we can't see what it was, but multiple reports have said that he stated this was an AIDS vaccine, which is not really true.
No.
So people are really worried about whether the parents knew the real risks here that were involved.
And the fact that, this will affect all the future generations of their children.
Yeah.
If they're even able to have children, which,
We don't know. He states, Professor He states that they had no unintended mutations, but...
Well, that's where right now we don't know, since this research has not been published in a peer-reviewed journal. So until it's published, until we can see how he did it and the results, there's only just conjecture we can make about if this worked or not.
Yeah.
But the majority of people, just based on how we've seen CRISPR work in the lab, we're like, this can't be right.
Right. And so Professor He actually did some work in the U.S. At Rice University, and his advisor at Rice University was apparently involved in consulting for this experiment. So he's now under investigations for ethics violations because of the part he may have played in the not so informed consent.
Mm-hmm. And this is really big within the science community because up to now, with a lot of these big gene editing summits, the scientists talk together and they kind of decide and kind of make their own laws, since the government's not really able to keep up with the science as quickly as it's moving forward. So for scientists to have made these laws and said we're not going to edit embryos, and even countries to have these laws as well, and someone to go and do it, it's just a major infringement on science as a whole. And it's causing a lot of people to look at all scientists and say, like, oh, are you all like this guy? Are you going to gene edit embryos? And what's the future going to look like? So it's like a huge step back for just science communication and transparency within science.
Yeah, I mean, with great power comes great responsibility to some extent. Like, we have a responsibility to regulate ourselves, and a lot of people are calling out Professor He's multiple advisors saying, did you know about this? Why didn't you step in here?
And one thing, he was not funded by the university he was at. He self-funded this study, so at least he wasn't using government money to do this.
Oh, and something I didn't really bring up when I was talking about the story is that all of this work was done in secret. So that goes back to the kind of the trust thing that we have in the science community. If you're not doing anything wrong, why did he feel like he had something to hide? And I mean, this can happen in fields where it's like a new technology and things are developing so quickly, and scientists don't want other people to find out about their work because they don't want them to steal their work. But the point this guy went to hide what he was doing is just outrageous. His university didn't even know what he was doing. Probably one of the most infuriating things about this story is that 2 days after Professor He came out with these news of these designer babies, his paper was published in a bioethics journal where he talks about how to do responsible gene editing, and he violated at least 2 out of the many principles he sets forth, one of which is to be transparent, which he wasn't, because he kept everything a secret, and still hasn't, published all the data, although apparently that's coming soon. And 2, he said in the paper that you should only use this technology for serious illnesses, and although AIDS is serious, it doesn't totally qualify because there are other treatments.
We've always said, we're a long way off from designer babies and that would never happen, but this is just evidence that it could potentially happen, which is really very scary.
So we need to find out where we're going to draw the line really fast,
yes.
and how to enforce that.
Yeah.